There's been a wave of media items that give equivalence to the weight of evidence for a lab leak and for emergence of COVID-19 through an intermediate host. But currently available data do not support that equivalence. That's the view expressed in a recent article written by the 11 international scientists who were part of a WHO China joint mission to look into the origins of COVID-19. What do the currently available data and scientific thinking tell us about the more likely natural origins of the virus? Recently, I moderated an international experts dialogue attended by 7 leading experts, convened by the Global Governance Institution, an independent think tank in China. And starting today, we're going to bring you in 3 installments what these experts said during the dialogue. We'll start with Wu Chong-yi, Chair Professor of the School of Life Sciences at Sun Yat-sen University. He's also former Chair in Ecology and Evolution of the University of Chicago.
The COVID-19 origin tracing not only is scientific,
we have to be very careful. The lab leak hypothesis.
To let science prevail.
I think I should say that I spent the great bulk of my career at the University of Chicago. I've been a professor at the University of Chicago for 30 years, 29 actually. So what I want to do, everybody say that of course we all understand the question of origin is a scientific question, but I want to emphasize the evolutionary perspective. I'm an evolution biologist. I have worked on the origin of flies, of dogs, and rice, simply because I use the evolutionary principles, and I think the virus is no different. So in the past, if we deal with something like the origin of SARS-CoV-2, we only have to demonstrate that natural process can reasonably lead to a product like that. But in this climate, we also have to go a step further and say that the natural evolution is the only possible way for such a thing to come about. And so this is really like we talk about the origin of universe, that people who have no background in astrophysics or physics in general could talk at length about the origin of the universe. We would find it very strange. But people who do not have anything, any background in evolution biology can speak about the origin of SARS-CoV-2. So a main question is not to show that natural process can lead to it, but actually to show that that's the only way to lead to it. And so the main argument is that this SARS-CoV-2 is extremely well adapted to human. And for something to become so adapted to human condition, it has to be a long and tedious process. For one thing, that the product has to be tested in human populations. It's just like if you try to release a driverless car to the society, you cannot expect that your first rollout, the car would perform wonderfully. It has to deal with also the road condition, anticipated or unanticipated, and you really work out the bugs, and maybe go through a long process. The driverless car can finally be adapted to the human societies. And the virus, if we look at it from a conceptual point of view, that it's unlikely that you can grab a virus in a sort of lab leak hypothesis, bring back to the lab, and maybe tinker with the 1 or 2 mutations, and then it would become such a pathogen. It's very unlikely. Let me give you the example. We have been looking at the sort of now very famous Delta strain, and we look at it, it actually has 31 mutations. Now, remember, Delta strain replaced the Alpha strain. It's a simple competition, it just has to do better. But for a virus, the origin of SARS-CoV-2 to be from adaptation to an animal host to human, I would say that 30 mutation would be a very, very low minimum. It probably would require at least 2-3 times that number. So the virus has to go through a very long process to become so extremely well adapted to human conditions. So the first thing I want to say is that it's inconceivable, either in a lab synthesis or to take a virus from nature, from any animals, and release it into human population and presto, and expected it to sort of burn through human population. I consider that extremely unlikely. And so what we have to do, there are two things that the first thing is that we have to define the conditions. We invoke the blind watchmaker argument that we published a paper, a group of 20 or so evolution biologists, including 7 or 8 of the most prominent one in China, that we define using the blind watchmaker argument to define the conditions under which that such a virus might emerge. And it involved a large population of animal hosts and a low density of human population, frequent contact, and so on, and herd immunity. So step by step it evolved. So I think this is important. I think in the last conversation with Madame Liu, I actually say that whoever wants to pursue the question of the origin has to propose a model. You cannot just say origin. And you want to, if someone wants to search for the origin, someone needs to propose a model. It's just like you want to catch a crime suspect. You at least have to have some ideas about the general profile of the suspect. You cannot just say suspect. You have to, and we actually defined it. Maybe you don't like this model and say, well, I don't think so. But everybody has to propose a model, what it looks like, what conditions under which this can emerge. And as far as I know, we are the only one who proposed an explicit model, right or wrong, we don't know. But if you don't have a model, you cannot start any search. It's meaningless. And I want to make another point that the new data, new evidence has emerged, and it's somewhat premature to say it now, that these new data will probably change drastically how we see this. For example, I mean, there are many not very credible reports about detection of SARS-CoV-2 in much earlier samples. But recently, there are 1 or 2 really believable reports that the variant has existed. And this is not the occasion, unless we have 2 hours for me to go through the evidence. I just want to say people should not be so confident about what the origin is. I mean, no scientist, I mean, a politician can be very sure about what the origin will be, but scientists should be more circumspect about that. So that's the first part. I think that's my view, evolution biology view about the origin of SARS-CoV-2. So I want to say something very quickly about the path forward. The COVID-19 is flaring up again. And and Delta strain really is a fairly dangerous strain. I think we should concentrate our effort. For example, the vaccination seem to be doing what we thought it could do. The social distancing has been tested, and treatment has been neglected. So we need to define a lot of things so we can resolve this problem. And so I would say that, let's face it, origin is not an urgent problem. If we solve we eradicate COVID-19, then we have decades to work out where the origin is and who should be responsible and so on for that sort of thing. But this is not the time to waste our energy, divert our attention to something of such a low probability.
Thank you so much, Academician Wu. Thank you. I hope we are able to focus more on the real science during this panel. However, we have to clear the noises out of the way, hopefully. Let me introduce our next speaker, Professor Shi Weifeng. He's Professor of Virology and Director of Shandong Key Laboratory of Etiology and Epidemiology of Emerging Infectious Disease of China. Since 2016, he has been engaged in scientific research studying the key molecules of emergencies and emerging diseases, including influenza A virus and Ebola. Professor Shi, the floor is yours.
Hey. Hello, everyone. Thank you for having me here. And my name is Weifeng Shi, and I'm from Shandong First Medical University. My group was involved in the discovery of the causative agent of COVID-19, and we have performing genomic surveillance of this virus in China and across the world. In fact, since the first reports of SARS-2 in December 2019 in Wuhan, China, there has been intense interest in understanding how this virus emerged in the human population. Recent debate has coalesced around the two competing ideas, a lab leak scenario and zoonotic emergence. Today, I would like to share the scientific evidence that may help clarify the origin of this virus. My first point is that SARS-2 is from nature is the consensus of global scientific community. Shortly after the identification of this virus, several world-known scientists analyzed the genomes of this virus and found that it had two unique genetic characterizations. First, there are 6 amino acid positions, which are extremely important for the attachment of SARS and SARS-2 to the human ACE2 receptor. Please remember these 6 positions because I will mention them later. However, SARS-2 is different from SARS at 5 of these 6 positions. In particular, SARS-2 has a higher binding efficiency to the human ACE2 receptor than SARS. Second, there's a 12 nucleotide insertion at the cleavage site of the spike gene of the SARS-2. This included 4 amino acids, PRRA. PRRA can be recognized by protease furin. Some scientists believe that this insertion could potentially enhance the pathogenesis of SARS-2. However, from the very beginning of the pandemic, there has been a conspiracy theory. Some politicians in Western countries believe in this hypothesis. They believe that the two unique genetic characterizations of SARS-2 are genetically manipulated. However, different groups subsequently reported several coronaviruses from pangolins. In particular, some of pangolin-derived coronaviruses share the same amino acids at those 6 important positions as SARS coronavirus 2. It should be noted that these wild pangolins were smuggled from Southeast Asia and were caught at the customer in 2019. In addition, 2 bat coronaviruses from Cambodia share 5 of the 6 amino acid residues. This evidence suggests that there are coronaviruses from wild pangolins and bats that are able to bind to human ACE2 receptor with very high efficiency. At the same time, from a bat sample collected from Yunnan in May 2019, we identified a novel coronavirus. This virus had a similar indel event at the cleavage site of the spike gene. Subsequently, a batch coronavirus from Thailand was also found to possess similar indel pattern. So although their insertion patterns are not exactly the same as SARS coronavirus 2, it suggests that such indel events can happen in nature. Just a short summary. So the so-called unique genetic characterizations of SARS coronavirus 2 have been found in coronaviruses from wild animals. Last month, 21 top virologists working on virus evolution published online a critical review. They contend that there's a substantial body of scientific evidence supporting a zoonotic origin of SARS coronavirus 2. Notably, these top virologists are from 7 countries, such as Australia, the USA, and the UK. These authors belong to 22 different affiliations. Therefore, SARS-2 is from nature is the consensus of global scientific community. This is my first point. The second point I would like to share is that there's no scientific evidence of lab leak. We also read through the reports supporting the lab leak hypothesis. However, most of them are preprints and are not peer-reviewed, so they are not reliable. Only very few of them are formally published. However, none of them is a research article, and none of them did provide scientific evidence to support their statement. Therefore, there's no evidence to support the lab leak hypothesis. While the possibility of a laboratory accident cannot be entirely dismissed, it may be near impossible to falsify. In July, a correspondence was published in the Lancet journal. The authors are a group of top virologists. They said that science, rather than speculation, is essential to determine how this virus reached humans.
Thank you very much, Professor Shi. Next, let me go to Professor Peter Foster from Cambridge, UK. I hope you can hear us now. Would you make a sign that you can hear me? Okay, you are still muted, but I hope you will be unmuted in just a moment. But I would like to welcome you and give our audiences a brief introduction of our next guest, who is fellow of the McDonald Institute for Archaeological Research of the University of Cambridge, UK. Now, Professor Peter Foster's research concerns the molecular population genetics of humans. He's currently Director of Research at the Institute for Forensic Genetics in Münster, Germany. He's also fellow of the McDonald Institute, as I said, and he's also an editor of the International Journal of Legal Medicine. So Professor Foster, the floor is yours, please.
Thank you very much. Let me first clarify, my research was conducted at Cambridge University, but I'm speaking in a private capacity.
Of course.
Okay.
Okay.
So thank you for the invitation to speak at this video conference. It's a pleasure. So my name is Peter Foster, and my teams in Germany and in Cambridge have since 1995 used a new type of genetic analysis method to trace and to date in absolute time the prehistoric human colonization of the world from an African origin in the past 60,000 years. So we succeeded in this by introducing a novel mathematical method, which we call a median network method. And this successfully clarified relationships between genomes, and for the first time. So in January 2020, when I first heard of the new coronavirus, I immediately realized we could trace the origin and development of the coronavirus using the same median network methods as in anthropology. So in our April 2020 paper in the Proceedings of the National Academy of Sciences of the USA, we identified the ancestral human coronavirus type by comparison with bat coronaviruses. We named this original human virus type A, and we also described a predominant descendant type, which we called B. Now, unbeknown to us, a Chinese research team at Peking University had already published a very similar result in March 2020, so 1 month before. And this is very good because it means an independent European team and an independent Chinese research team have reached the same conclusion. So that is very satisfactory in science. So using this analysis, we distinguished 3 levels of origin of the virus. At the earliest level is the transmission of the coronavirus from animal to human. As we've heard in the previous speakers, the closest known bat coronavirus differs little from the current human coronavirus by only 4% of the genome. And that corresponds to a maximum of 50 years of separation according to the mutation rate we measured. Now, these 50 years is only a maximum estimate because we probably have not yet found the most closely related bat population, as the previous speaker indicated. Furthermore, the bats and the first patients live in the same geographical region, so the animal-human transmission must have happened quite recently, in my view, possibly as recently as 2019. And the exact transmission mechanism is under investigation by Chinese researchers for some years, in fact. So in 2015, the team of Xi Zhengli and colleagues surveyed villagers in Yunnan province who were living near bat caves, caves full of bats. And this team found that 3% of these villagers had antibodies against bat coronaviruses. And this evidently was occurring through natural transmission from animal to human. The second level concerns the geographical origin and spread of the symptomatic virus within China. So what I did, and also for this talk, I examined available genome sequences, and I also examined clinical histories of early patients. In this map, this shows where the original ancestral A types and the derived B types were detected in patients until mid-January 2020. So the patient numbers are very low at this early stage. The black pie slices are the original A type, and the white portions are the derived younger B types. So although we have very few samples at this very early stage until mid-January 2020, so only 34 B types and 7 A types, it is clear that the A types are very rare in Wuhan and in Hubei province. Only 1 in 29 patients have the original A type. In contrast, there are more A types detected in southern China. So this makes Wuhan a less likely source. Now in the next map, this is 1 week later. The sample sizes are still very small. So now we have 61 B types and 22 original A types. But it is clear that the A and B types in this week later are spreading in parallel across China. Remember that Wuhan had mainly B types up to the previous week. And this suggests that Wuhan is not the major source of the epidemic. If Wuhan had been the source, we would have expected a spread of only or mainly B types. But instead, what we see here is a mix of black and white spreading, a mix of A and B types spreading. So this is not easily explained by a spread from Wuhan. At the third level, we can ask where the current global variants come from. And these global variants, we all know, alpha, beta, gamma, delta. In our original network analysis in April 2020, we had noticed that B types, including a so-called B1 type, were behaving aberrantly. So you can see that in the statistical analysis, this B1 type is behaving differently. And that is predictive of the rapid spread of B1 in 2020 from Asia to Europe and then worldwide. Several months later, the Los Alamos National Laboratory in the USA confirmed that this B1 type causes a higher viral load in patients than non-B1 types. Our network analysis can therefore be a valuable tool for guiding clinical research. So you can see at an early stage statistically that a virus is behaving or a variant is behaving differently. So I thank you for your attention. And if you are interested in a fuller account of our research, see the website of the Cambridge Philosophical Society, where I have a longer lecture on the subject.
The COVID-19 origin tracing done is scientific.
We have to be very careful.
The lab leak hypothesis.
Let science prevail. The last speaker was Dr. Peter Forster, Director of Research at the Institute for Forensic Genetics in Germany. And that's the end of part one of an international experts dialogue brought to you by the Global Governance Institution in collaboration with The Point on CGTN. In the next episode, we'll hear from an epidemiologist from Harvard Medical School, Dr. Eric Fagel-Ding, Professor Luis N. Juarez, an international member of the National Academy of Sciences of the United States, and Wu Wei, professor at the Nanjing University Public Health Research Center.